Hello, I am Dongyoung Kim, an undergraduate student at Aji University. Today I am going to talk about my ongoing research. The topic of the presentation is the differences between cave surface populations of cryptic harassment endemic to South Korea. So I will begin my talk with an introduction to troglomerf. Even though caves exist in numerous regions around the world and there is a wide variety of taxa in those caves, cave-adaptive organisms show common traits. We call this troglomerf, and such features include depigmentation, R reduction, and appendage elongation, which are morphological traits that are arisen from convergent evolution in the cave environment. This is my research subject, coreanus. The upper one is a sample of the cave population, and the bottom one is from the surface population. For the cave population, you can see troglomerfs such as depigmentation and long appendage. One of the ultimate questions in this study is how the troglomerphism in coreanus evolves, whether it occurs among lineages or within a lineage. Genus Kaolinonychus is a member of family Paranonychidae, Elaniatoris, and in this genus there is a monotypic species coreanus. In 1966, Japanese scholar Seisho Suzuki described K. coreanus with two subspecies, K. coreanus and K. coreanus longipes. I believe advanced research is needed in this taxonomic system for two reasons. The first reason is that this classification does not reflect ecological features, and the second is that it does not encompass the individual variations in the key characteristics between the two subspecies. Looking at their habitat, K. coreanus longipes were collected from only one locality, Gongi Cave, while many recordings of K. coreanus have been found through several caves and surfaces environments. The current classification system appears to consist of longipes and the rest, so I think that the revision on this text should be conducted and a comparison between cave and surface groups should be established first. Therefore, I conducted extended research on the phenotypic and genetic variation among K. coreanus populations that varies in their habitat between cave and surface. To figure out the differences between them, I chose three characteristics to investigate. First, we compared the length of appendages among the populations. Next, I compared the body shape using geometric morphometrics. And lastly, I analyzed the data from a preliminary genetic experiment on mitochondrial DNA. Regardless of the previous classification, I divided collected specimens into two groups according to their habitat. We sampled 22 males from each habitat. These are collection sites. Almost every sample was collected in Gangwon, Chungcheong, and Gyeonggi province in Korea. For the length measurement, I compared the body length, the second leg length, the pedipalp length, and the proportion of segments in the appendages. The second leg is known as the appendage that acts as an antenna in harvestment, so it is considered to be important when adapting to the apodic environment. On the other hand, pedipalp is used as a raptorial leg in the rhinoceros harvestment. Thus, the variation in two appendages between cave and surface populations may provide evidence of whether the phenotypic variation is facilitated by the adaptation in the cave populations. I divided the length of the appendages by the body length for the following analysis. Afterward, I also measured the ratio of each segment to the total length of the appendages. Next, I carried out landmark-based geometric morphometrics analysis to see the shape differences. After setting 17 landmarks on the lateral view of the skull with the TPS util and TPS dig, I conducted a generalized procrustes analysis. And using generalized data, we did a multivariate analysis using MorphoJ on R package geomorph. Now, let's see the result. Here is the principal component analysis result for all lengths and ratio data. Looking at the plot, you can see that two habitat groups are divided by the PC1 axis. And we can know that the high contribution variables of PC1 include second leg ratio, second leg length, pedipalp length, etc. So let's check each variables in details. As compared to the non-significant variation in body size, there are clear differences in the ratio of the second leg and pedipalps. It means that the cave populations have larger second legs and pedipalps relative to their body than the surface populations. In particular, the difference in the second leg is more distinct between the two groups than that of the pedipalp. And at the segment, the ratio of trochanter, patella, and tarsus in the second leg shows significant differences between cave surface populations. Interestingly, the segment close to the body, such as trochanter and patella, are longer in surface groups. On the other hand, the most terminal segment tarsus shows a significantly larger ratio in cave groups. Next is the analysis of the shape of the lateral scrotum. When I saw this PCA result, I thought that the cave groups were clustered, but there exist many overall regions. Therefore, it is considered that there is just a slight difference between cave surface in scrotum shape. Nevertheless, to figure out the subtle difference between the two groups, we conducted orthogonal partial least square discriminant analysis. According to the variable important projection, VIP scores, we can assume which landmark variables make the two groups different. Among the variables with high VIP scores, I selected ones that are significantly different between the two groups in statistical tests. And applying them to each coordinate, we were able to estimate the shape differences between them. So here are the magnified models that reflect the differences in the scrotum shape. We can see that cave populations have lower heights and wider lengths than surface populations in the shape of the lateral scrotum. Next is the result of the pilot study on molecular phylogeny using cytochrome C oxidase 1 region. When we mark them with a habitat, we can see that there is no strong association with habitat. On the other hand, some cohesiveness is identified geographically. In conclusion, there is a difference between cave surface groups in appendage ratio and length, especially in the second leg. And the morphological data show that the existing classification does not reflect the wide spectrum of individual variations. Next, the ratio of the most apical segment in the second leg is larger in cave populations than that of surface populations. In the scrotum shape in the lateral view, the cave populations have relatively low and wide shapes. Analysis of mitochondrial DNA C1 region confirms that they are more geographically related than ecological factors. Through the integration of morphological and genetic analysis here, we could assume that the cave adaptive characters in Kaolinonychus coreanus are due to parallel evolution in each geographical group, rather than synapomorphia of the early divided lineage of clade. From this, we could pose the requirement of revision from the taxonomic system by Seisho Suzuki. Now, I would like to discuss some points that need consideration. Firstly, the presented study revealed the necessity of advanced taxonomy research. According to Suzuki's review paper in 1975, the two subspecies are identified with a second leg ratio. If the ratio is 3, then it will be coreanus coreanus, and if the ratio is 6, it will be longipes. However, based on result of this study, it was observed that there were many continuous variations between 3 and 6 in the second leg ratio. So I think this classification character or these taxonomic systems need to be modified. I hypothesized that K. coreanus could be divided into 3 forms. In my hypothesis, there is an intermediate form between cave surface populations, and the intermediate form may provide some gene flows between cave surface populations through conservative genital morphology. And it is noteworthy that the intermediate samples in this bar plot are highly correspondent in the cluster when partitioning around the midwich were carried out with all length data. So the advanced study will focus on the question that does intermediate groups really exist and how the gene flows occur among the geographical groups. If I develop this study further, the first thing to do is to get more samples from more regions and get more various somatic data. And because their bodies are stereoscopic, there were limits of 2-dimensional geometric morphometrics to treat error among samples. If we can conduct a 3-dimensional geomorph, we will be able to get more reliable data. In molecular phylogeny, CO1 genes alone have limitations in revealing their intragroup relationships. So it is expected that more interesting research will be possible if a population genetic approach is added. Through further studies, we will keep trying to approach the answer to the question, how did they evolve and what is their relationship? Thanks for watching.